Diabetic kidney disease is a major public health problem in Singapore. In the world, Singapore has the #1 incidence of diabetes as a cause of end-stage kidney failure.
Every day in Singapore, 9 persons will be diagnosed kidney failure. So every day there will be 9 people in Singapore who will require a kidney replacement therapy.
With Dynamo, we're bringing together a team of scientists, people working both in basic discovery science as well as clinicians working with patients with a single focused goal of helping patients who have diabetic kidney disease. It's happening not only here in Singapore, which is where the project is really focused, but also in Europe and in the US.
Dynamo is a very large, ambitious project which provides a multidimensional observation from genetics to patients, from animal model to cell-based model, to study and understand diabetic nephropathy.
Dynamo will be very powerful to enable us to discover new biomarkers, identify people at risk of diabetic kidney disease. It will also reveal to us in depth the disease biology.
In Dynamo, we express the goal to reduce the incidence of end-stage kidney disease caused by diabetes in Singapore by more than 25%. So we hope that these studies will have real impact on patients and their families.
In Dynamo, we have access to several large prospective multi-ethnic cohorts. And from these cohorts, we have collected their baseline biological sample, and the participants are observed over a decade. We organize our patients into various subgroups, those who experience progressive diabetic kidney disease versus those who have stable kidney function. And from the baseline sample collected, we can observe a metabolite signature to predict who is going to experience kidney progression.
In this study, there are 1,000 patients with diabetes. We sequence the whole genome and then we try to find changes in the genome that correlate with complications of diabetes or absence of complications. We focus first on mutations in the parts of the gene that encode for proteins. Then we have a gene-level analysis where we can study individual genes and find out which mutations are there. And then the third is then to study clusters all over the genome which are enriched in individuals that either have complications or who do not have complications.
We hope to be able to identify aberrant pathways in patients with diabetic kidney disease who are fast progressors and determine whether or not these are druggable targets. Novel biomarkers may improve the performance of risk calculators by maybe 10–15%, but by not that much more. Hope is that transcriptomics, proteomics, metabolomics, in a serial manner will be able to actually produce a risk factor analysis or to diagnose diabetic kidney disease or even prognosticate diabetic kidney disease. This, coupled with bioinformatics and artificial intelligence, should be able to bring us very close to 90% in the future for novel biomarkers.
Diabetic kidney disease has been a subject of research for many years, and despite that fact, we don't really understand the major root causes of kidney disease and diabetes. We can take advantage of genetic manipulation in our mouse models to very precisely manipulate pathways that may have functional importance in causing diabetic kidney disease. We've found metabolic abnormalities that are similar both in humans and in our mouse models, which we're trying to use productively to get at basic causes of kidney disease and increased risk.
I'm a stem cell researcher. In my lab, we are particularly interested in generating three-dimensional kidney organoids from human pluripotent stem cells in order to allow us to model some of the features in diabetic nephropathy. In diabetic nephropathy kidney patients, VEGF-A signal is very badly disrupted. Podocytes are the major source of VEGF-A within kidney. Currently, we are trying to employ chemical reagents which will cause podocyte toxicity to see whether this will affect VEGF-A production, which will cause reduction of vessel network within the kidney organoids.
In diabetic patients, we often found that diabetic nephropathy and retinopathy occurs together. We hope to use retinal image to predict the risk of the development of diabetic nephropathy. In my lab, we mimic vascular abnormalities. We found that dysregulated lipid metabolism and mitochondrial dysfunction are common for patients with both diabetic retinopathy and nephropathy.
For me, the most exciting part of the research in Dynamo is the opportunity to bring together teams of very talented scientists that are dedicated to addressing what is a major public health problem in Singapore and around the world.
We have the opportunity to work with a diverse group of colleagues, from basic scientists to clinicians to biostatisticians. There's this shared passion of trying to find solutions to help people with diabetic kidney disease that keep us going.